Microbiology video part 2 for chapter 23, Brock. Part 2, pathogenesis. In this chapter we'll talk about pathogenicity and virulence, adherence, invasion, infection and virulence factors, exotoxins, and endotoxins. Pathogens use a variety of strategies to establish virulence. Virulence, as we've talked about previously in class, is the relative ability of a pathogen to cause disease and can be quantified. The entry of the pathogen into the host looks something like this, where pathogenesis is the ability of microorganisms to initiate disease, so entry, adhesion, invasion, infection, and then some form of either toxicity or invasiveness leads to tissue damage. This figure basically is an overview of everything we're going to talk about for the rest of this chapter. You can measure virulence a number of different ways, but remember we defined it as the relative ability of a parasite to cause disease. Among measurements for toxic compounds or even bacteria or viruses, like virulent microbes, one of them is called the LD50, or lethal dose 50th percentile, which simply means the amount, or in this case the number of bacteria required to kill 50% of the test organisms in a group. Different organisms exhibit different virulences depending on the subject being infected, i.e. salmonella in a mouse has a different LD50 than the same strain of salmonella in a human. That said, we can extrapolate from animal models to approximate LD50s for human models. For human cases. Pathogenic organisms that are kept in the lab and not passed through animals at all often lose their virulence, and that is said to be attenuated. Highly virulent pathogens show little difference in the number of cells required to kill 100% of the population as compared to 50% of the population. In this example, you have a highly virulent Streptococcus pneumonia strain on the left and a less virulent Salmonella typhimurium strain on the right. You can approximate the LD50s by looking about where 50% of the test subjects are killed. This number is somewhere between 10 and 100 cells per animal for Streptococcus pneumoniae in this situation, but somewhere between 1,000 and 10,000 cells per animal in Salmonella. So this is the higher virulence. After establishing virulence, you can determine what mode of virulence occurs in general terms inside an animal. And in general, there are two ways that bacteriums or microbes can cause damage to hosts. One is by releasing toxins into the cell, causing disease by means of a toxin. And two is by general invasiveness, growing in the body and disrupting tissue through growth. Usually there's a combination of both toxins and invasiveness, but there are extremes at either end. If you go back to the original figure that we showed at the beginning, you'll see that before anything can happen, we have to have adherence. This is the same for viruses, bacteria, yeasts, anything that's going to cause infective growth. There has to be attachment. So bacteria and viruses that initiate infection have to adhere specifically, usually to epithelial cells or through a break in the skin. And these interactions between molecules on the surface of the pathogen and the host cell are key to the initiation of infection. On the left, we have attachment through capsule layers. There's E. coli in the capsule layer. And on the right, we have Vibrio cholera attachment without a capsule layer, where there'll be specific interactions between proteins on the cells. Either way works fine, because these are mucosal membranes. So if you have a nice gooey capsule, it's going to stick to the mucosal layer on the epithelial cells and the villi in the epithelial cells. Just like you'll have specific attachments between proteins on the villi and the bacterium as well. Some of these organisms will exhibit tissue specificity, only attaching to certain tissues and infecting them. And then some, like the viruses, will display host specificity, where one strain of bacteria is more likely to infect one animal over another. I saw this in my research with turkeys. When we started isolating Staphylococcus from turkey beets, we found that the Staphylococci species that were in the turkeys looked more like the Staphylococci species you might find in some companion animals like dogs or cats versus the Staphylococci that you would find in a human being. That said, I still have been able to isolate Staph aureus from turkey beaks. So some of it's specific, some of it's tissue specific, and some of it can be host specific. Oops, back up here. Somehow this got deleted. Occurrence is going to be occurring through either the capsules and slime layers, or extracellular macromolecules, or fimbriae and pili that are sticking out of the bacteria. It's on this slide. So often pathogenic strains of a bacteria such as E. coli have fimbriae that recognize and bind up epithelial cells in the intestines in order to cause disease. That way they can recognize these cells and attach to them. Here's colonization. Excuse me. Here's colonization of capsules surrounding strep pneumonia. And here are the fimbriae sticking out of an E. coli. If the fimbriae sticking out of the E. coli have specific proteins that will attach to epithelial cells, say on the intestine cell wall, they're called colonization factor antigens, or CFAs. Here's a table summarizing the different adherence factors. Capsule and slime, adherence proteins, like CFAs, lipoteichoic acids, like in gram-positives, can also attach to cell material, and fimbriae, and the pili, CFAs here. Most pathogens penetrate the epithelium to initiate pathogenicity. This process is called invasion. Growth can occur at the site of penetration, in the submucosa, and on intact mucosal cells as well. Growth may also be established at distant sites from the point of entry via the blood or lymphatic systems. Here, E. faecalis is shown infecting the blood and tissue of a patient. I'm recording. Infection is any situation, oops, hold on one second. The availability of nutrients is most important in affecting pathogen growth. Pathogens may grow locally at the site of invasion or may be spread throughout the body. Most pathogens must grow to cause disease. This process is known as colonization. Colonization depends on the local pH, nutrient availability, and the reduction potential or oxygen content. Not all nutrients are available to all pathogens, such as sugars and other growth factors. Some virulence factors are just designed to take nutrients from a body and provide them to another cell. So now we can sort of look back at our definition of infection and talk about it as any situation in which a microorganism is established and growing in a host. Here it's defined as not a member of the local flora, but it can often be a member of the local flora. I'll just take that out. Infections are often localized with a focus of infection nearby to where they gained entry to the body. They can travel via the lymph system or the bloodstream and become systemic, and then you have a big problem. Generalized infections usually start locally, and systemic infections are very dangerous. The term bacteria anemia is the presence of bacteria in the bloodstream where they shouldn't be, and septicemia is the blood-borne systemic infection. Septicemia can be dangerous because of overstimulation of your immune cells that can lead to massive inflammation, septic shock, and death. The things that allow bacteria to cause these types of infections and colonize are called virulence factors. These are compounds that are coded for on genes that enable a microorganism to establish itself on or within a host of a particular species, enhance its potential to cause disease. We can assess virulence by looking at both invasiveness and toxicity, as I said before. For example, Salmonella species encode a large number of virulence factors. Several groups of genes that in direct invasion are clustered together on the chromosome as so-called pathogenicity islands. Another island of pathogenicity on Salmonella contains genes that promote a more systemic disease, and still others promote hiding from the immune system. In addition to that, Salmonella contains resistance plasmids that make it resistant to antibiotics, or can contain that. In this picture, you can see a number of different virulence factors, some of which we're going to explain in some detail. Salmonella has an endotoxic LPS layer around it that causes the body discomfort when lots of these cells are grown up and die and the endotoxin is released. They also have enterotoxins that can cause diarrhea, which are proteins that are excreted out of the cell. Cytotoxins that inhibit cell protein synthesis. A capsule called a V capsule that inhibits complement binding, which is part of the immune system, which we'll talk about later. Flagella add motility and also possess an H antigen which enhances adherence. O antigen inhibits phagocytic killing by the immune system. And cytophores sequester away iron from cells. Salmonella establishes intracellular parasitism as a mode of infection versus growing outside of tissues. They grow in the cells themselves. So when they do this, they can avoid being eaten and destroyed by macrophages, specialized white blood cells. They neutralize the tools the macrophages use against them. Thus, Salmonella has a bag of tricks to add to its virulence, and is very typical of a pathogen having more than one type of virulence to try and grow. We can classify all these virulence factors as either enzymatic or as exotoxins. Enzymatic virulence factors are enzymes that aid in the infection and transmission of disease by breaking down cell components and making cells vulnerable to infection. Examples include hyaluronidase, made by staph, strep, pneumococci, and some Clostridia. This enzyme breaks down hyaluronic acid, which holds cells together in tissue and allows further infection from the focus outward into the body. Proteases, nucleases, lipases all break down macromolecules, collagenase as well. Streptokinase is used to break down blood clots and facilitate infection. And before there was widespread use of other blood thinners, streptokinase was actually used for heart attack patients to break up blockages. And then, of course, coagulase is the one we tested for in the lab, which is indicative of a staph A infection. These are all enzymatic virulence factors. They differ from the exotoxins. Exotoxins are also made out of proteins, but they don't work enzymatically. They're made in the cell and then released outside it as it grows. There are three basic types of exotoxins, two of which we'll talk about in this chapter. The cytolytic toxins work by attacking cell constituents causing lysis, like the hemolysins we saw in lab. And the AB toxins, which have two covalently bonded subunits. These toxins have one protein or subunit that binds onto a cell receptor and then allows the other subunit to cross over the membrane and cause damage to the cell. Superantigens are a third type of exotoxin that can elicit strong, non-specific responses from your white blood cells in the immune system. And we'll talk about those when we talk about immunity later on. So cytolytic toxins work by degrading cytoplasmic membrane integrity, causing cell lysis and death. These can be toxins that lyse red blood cells like we saw in the lab, so we call them hemolysins, right? Or they could be white blood cells, in which case we call them leukocytins. An example of cytotoxin is staphylococcal alpha toxin, which kills nucleated cells and lyses red blood cells as well. The staphylococcal alpha toxin binds to the lipid bilayer, or 7 identical subunits, for a heptamer. Once the heptamer is formed, it changes shape to form a membrane-spanning pore. The pore disrupts the cell, and the cell dies from osmotic pressure and loss of metabolism. This is sort of a typical type of cytotoxin reaction. Some of these hemolysins can attack specific phospholipids called lectins, and so they're called lectinases or phospholipases, right? Examples that do that are from the Clostridium perfringens, as an alpha toxin does that. Some hemolysins are not phospholipids. They can be streptolysins, which attack sterols in the plasma membrane, and still other bacteria produce leukocytins, which I mentioned before, which lyse white blood cells. This one forms pores in red blood cells and nucleated cells. Here's a picture of it on the blood plate, just like we observed in lab. The exotoxins are primarily outside the cell, and there's two general groups. The AB toxins, which we'll talk about in depth, and then also the endotoxins, which we mentioned at the beginning of the course when we started talking about LPS, so gram-negatives. So as we said before, the AB toxins are made of 2 protein subunits. The B subunit binds to the host cell receptor and then transfers the damaging part of the protein into the cell membrane. Examples include diphtheria, tetanus, botulism, and Shiga toxin, which we'll talk about in lab next week. So this is a picture of how diphtheria toxin works. The AB toxin is released, and the B subunit will attach to the membrane, allowing the A subunit to come in. So the protein gets cleaved and the A subunit goes in. The A fragment in diphtheria disrupts protein synthesis by blocking amino acid transfer to ribosomes on tRNA. The A toxin attaches an ADP ribose molecule from NAD to an elongation factor required by the ribosome. The modified elongation factor no longer promotes the binding of tRNA, and protein synthesis stops. So the cell dies. The interesting thing about Corynebacterium diphtheriae is that the bacteria does not produce the toxin until it has been genetically transformed by lysogenization. So the genes for this toxin were originally on a virus. So a specific bacteriophage must infect C. diphtheriae and place the genes for the toxin inside the bacterium to make it virulent and pathogenic. Another interesting thing about diphtheria is that iron acts as a negative control of gene expression in transformed cells. So when iron levels are high, right, in other words, the bacterium can get all the iron it needs, the bacterium does not produce the toxin. However, when iron is in short supply, the bacterium produces the toxin and kills cells, which then release all their iron. Another example are the AB toxins made by Clostridium tetani and Clostridium botulinum, who produce some of the most potent bacterial toxins known to man. The botulinum toxin consists of several different proteins, AB toxins, that are the most potent biological toxins known. Tetanus toxin is also a known AB protein neurotoxin. Clostridium botulinum rarely grows directly in the body, but can grow in preserved foods. The botulism toxin is the most toxic biological substance known. The toxin is a series of 7 related AB toxins. One of these is toxic to the central structure that reacts with the non-toxic AB proteins to form the activated toxin. The complete toxin can then inhibit the release of acetylcholine from terminal motor neurons, causing flaccid paralysis and death. So at the normal neuromuscular junction, an electrical signal gets sent down the nerve. It releases the amino acid acetylcholine, which binds to muscle tissue and it allows your muscle to contract. In the presence of botulinum toxin, that release, no matter what signal is coming down here, that release is blocked and your muscle does not get any signal to contract and you get flaccid paralysis. At least 2 of the 7 AB toxins that make up the final protein are coded for on lysogenic bacteriophages specific to this bacterium. If you get enough of this toxin in your blood system, it can actually paralyze your diaphragm and you die from asphyxiation. This toxin is also the reason babies aren't allowed to have honey until they're a couple of years old. Botulinum doesn't grow very well inside the gut, but it is an anaerobe. And if the gut hasn't got an established microbiota or natural flora, that like when you're a small baby, it could in fact grow inside an infant. And when that happens, babies can get poisoned. They don't eat the honey, or you're not allowed to eat the honey, because the bees carry the pollen to make the honey, and they get contaminated. They can get contaminated with botulism spores. Doesn't affect you if you have if you're an adult. The tetanus toxin, another AB toxin, is formed when C. tetani grows in anaerobic wounds. The bacterium does not invade the body from the site of infection, but the toxin can spread through neural cells. Tetanus toxin is an AB polypeptide that blocks the release of the neurotransmitter glycine from inhibitory neurons. Glycine acts to relax muscle tissue by blocking the release of acetylcholine. So here's our stimulatory system with the muscle tissue and the nerve that stimulates it. Once the signal is turned on, it has to be turned off, and that occurs through another nerve system that uses a different amino acid as a neurotransmitter, glycine. So when the glycine is released, when you want to relax your muscles, it then tells this nerve to stop releasing acetylcholine, and the muscle relaxes. If tetanus toxin is in the area, it blocks the release of glycine, so the muscle gets turned on, but then can't relax, and you get spastic paralysis. Lockjaw is the tetanus-induced paralysis of the mouse muscles, and if respiratory muscles are involved, death can occur. Neither one of the, while tetanus grows in the wounds, in anaerobic wounds, it's not invasive, and all the damage it does is through traveling of the toxin up the nerve pathways. Neither one of these is very invasive at all, just very toxic. Another class of exotoxins are the enterotoxins. Entero meaning gut, right? So these are exotoxins that activity affects the small intestine. They generally cause massive secretion of fluid into the intestinal lumen, resulting in vomiting and diarrhea. The example in the book is for cholera. Cholera toxin is an AP toxin that attaches to a specific glycolipid of the epithelial cytoplasmic membrane. After attachment, the A chain activates an enzyme in the epithelial cells that transforms ATP to cyclic AMP. Cyclic AMP acts as a hormone inside the cell, and the cell pumps and signals the cell to start pumping chloride and bicarbonate ions into the intestine. This causes the ionic balance to be disrupted, and the cells respond by flooding the lumen with water. This reaction is so violent in cholera that water is lost faster than it can be taken up, and a massive net loss of water occurs. And cholera victims usually die of dehydration unless they can be hydrated aggressively. Other enteropathic bacteria have similar toxins to cholera toxin, perhaps not as violently or as strong. For example, both E. coli and Salmonella species have similar modes of action. A third form of toxin, which is not an exotoxin, is called an endotoxin. Endotoxins work. Endotoxins are called endotoxins because they exist within the LPS of gram-negative bacteria, that lipid A fraction that we talked about in the beginning of the course. The toxicity of endotoxins is less than that of exotoxins, though one could conceivably die from exposure to enough endotoxin. The toxic part of the LPS layer is the lipid A, and the core polysaccharide is necessary to obtain a toxic effect with the lipid A. These toxins can cause fever, diarrhea, inflammation, and a general lowered immunological state. Here's a table comparing the properties of the exotoxins and the endotoxins. Exotoxins are proteins that are secreted by active cells. Endotoxins are part of the LPS that gets released after the cell dies. These are very specific and highly toxic. These are very general and low to moderate toxicity. These are highly immunogenic, so you can be stimulated to create antibodies against the toxin and protect yourself. These are relatively poorly immunogenic, which means you don't mount such a big immune response to them. What else? These are often on plasmids. These are often on chromosomes because they're structural. The end of the chapter talks about the beginning of resistance and the innate properties of resistance in the body. When something's innate, it's there from the beginning. So these are non-specific barriers to infection in the body that do not recognize highly specific pathogens, but rather are generated all the time to block infection. Host defenses can be split up into groups. Specific host defenses that target an individual species and non-specific defenses that target a wider range of invasive pathogens. Here we're going to talk about the non-specific ones. Host natural resistance is conferred by many different factors. As we talked about before, some species are not even affected by certain pathogens, and many animal pathogens are not human pathogens. Cold-blooded pathogens rarely attack warm-blooded animals. First and foremost, structural integrity of tissues is a barrier to infection. Skin and mucosal tissues, when they're intact, prevent attachment and infection. So intact surfaces usually prevent colonization. Damaged surfaces promote colonization. Skin also secretes acids at lower pH, which inhibits growth. The The second physical barrier would be the cilia and the mucosal tissue that causes pathogens to be expectorated or swallowed, like we talked about in class today. Acids in the stomach that act as a barrier, right? And then finally, if something gets past the acid in the gut, you have the normal flora that usually outcompete any organism that made it that far. So if the organism can't attach and can't grow, there's no infection. In addition to that, there's enzymes in your tears and in your blood and saliva, such as lysozyme, that break apart cell walls of bacteria. Blood itself has a protein called beta-lysin, which can destroy microbial cells. It's there all the time. You're born with it, so it's innate. Pathogens are tissue-specific, as we've said before, so one type of avenue of infection does not fit all parasites. When a host becomes compromised, it means that the host has lost one or more of the defense mechanisms previously mentioned. Hospital patients are often compromised hosts. Many procedures are invasive and leave avenues open to infection and colonization. In addition, the stretch of surgery diminishes resistance in patients. Compromised hosts exist outside the hospital as well. Smoking, no sleep, alcohol and drug use, and poor nutrition all contributed to lowering resistance to pathogenic bacteria and viruses. Infection by other diseases such as HIV compromise the host further. Things that can affect, or factors that affect making the host more or less susceptible as far as innate barriers, and can include things as age. Infectious diseases are more common in the very young and the very old than they are in the middle-aged. The young do not have immune systems or natural flora to impede infection, and the old no longer can mount as good a counterattack with their immune systems if one does start. Stress can predispose a healthy individual to disease as well. How many of you know someone who always gets a cold when they're stressed out? Stress increases the anti-inflammatory hormones and removes a natural barrier to infection. Diet can also play a role in host susceptibility to infection. People who do not have the required nutrients are susceptible to infectious disease. One only has to watch a TV or look to see where in the world infectious diseases are rampant, and there's strong correlation there in countries where people do not have enough to eat. Also, dental caries. Remember the example of dental caries at the beginning of the chapter? This is an example of where diet allows infection to take place. Finally, certain genetic conditions can compromise a host. There's numerous immunodeficiencies, some of them very drastic, but some of them very minor, and just appear as differences between members of the population. Some of them, for example, if they're in B cells, would render the host very susceptible to bacterial infection, while others, if they were in T cells, render the individual more susceptible to viral infection. That's the end of this chapter. I hope you enjoyed the video.